So today we're talking about, drum roll please.
Why are we doing this one again?
For the ratings.
I thought we just needed A on this project, not the ratings.
Play the drum roll. DNA replication.
Save me, G-Bus.
So DNA replication is the way that your genetic material is passed on in your somatic cells. Your somatic cells are any of your cells that are not your sex cells. This process creates various traits and proteins and eventually makes up your phenotype. Genes are made up of two alternate forms of those genes, and those alternate forms are called alleles. All of your genes together make up your genotype. We need to talk about the structure of DNA, which is one of 3 nucleic acids. The other ones are RNA, and that creates proteins, but we'll talk about that later. And the other one is ATP, and that helps you do your cellular functions. Now, there are 4 bases, and adenine pairs up with thymine all the time, and guanine pairs up with cytosine. Now, adenine and guanine are your purine bases, and thymine and cytosine are your pyrimidine bases. So, Rosalind Franklin discovered that DNA was a double helix, and the way that she did this was through something called X-ray diffraction. But Watson and Crick stole her findings. Well, they didn't really steal it, but we're saying they stole it because she did it. But so they stole the shape of DNA from her, and they made a model to represent the DNA. And what did they win? They won a Nobel Prize. But what did she win? She won a grave. But hey, yeah, that's definitely fair.
Stupid.
Yeah, I'll allow that because it is stupid. They even thought that DNA was a triple helix. I mean, come on. So your nucleotides are made up of 3 main things. One of them is a pentose sugar. So that's a sugar with 5 carbons. And then you have your nitrogenous base, and you also have a phosphate group. DNA helicase is the molecule which is responsible for breaking apart the hydrogen bonds that glue those nucleotides together, and that forms a replication fork, which allows your DNA to replicate.
Like when you build beautiful house of cards, right? You take so many cards and you take a lot of time to do this one, right? Okay? And you look at it finally and you're like, oh my god, it's so beautiful. You're so proud to take selfies with it, to take pictures of it, you post to Instagram, but then your one friend puts coin on it and it breaks, okay? It breaks. And now you're disappointed, okay? Now you're very mad, okay? I think Stephanie did this one once. Okay?
Hold on. First off, that was Elena, okay? And second off, she was just
testing the structural integrity of it, okay? Alright? Structural integrity, my butt. Okay, I got the aesthetics. You know what? In a time that you could not even complete 1 castle successfully, I made 3, alright? So I don't want to hear about it. Anyways.
This splits the DNA into 2 strands, the leading and lagging strands. The enzyme primase creates a primer, which attaches itself to the end of the leading strand. The other strand will be the lagging strand, but we'll be talking about the leading strand during this section. So, the primer attaches to the 3 end of the leading strand, marking the beginning of DNA synthesis. DNA polymerase is an enzyme that binds to the leading strand and adds complementary nucleotide bases. This happens in the 5 to 3 direction.
Yeah, all this talk about the leading strand, but what about the lagging strand, huh? We can't forget about this one. That's a good question.
You remember the primers I mentioned earlier?
Jace.
Well, the primase enzyme binds to the lagging strand in order to create the primers. Okazaki fragments, which are chunks of DNA, are added to the lagging strand in the 5 to 3 direction.
Great, but how do we get the new DNA?
Gosh, you're so impatient. You asked the question in the first place.
Oh, we are already 2 pages in the script and we need to talk about DNA replication, okay?
But we never stopped talking about replication.
Okay, fine, continue.
Whatever. So, once the nucleotides are paired, exonuclease strips away the primers, leaving gaps which are filled by more nucleotides. Polymerase proofreads the DNA strand, and ligase seals the sequence into 2 double strands.
Does that mean that for one strand, 2 are formed? Exactly. Oh. And each strand has 1 strand and a new one? Right again.
And that means that there are a variety of different enzymes that are used in the replication of DNA?
You're really getting the hang of this.
But I thought enzymes break down molecules, not build them.
Well, enzymes are used to break down substrates, but they can also use small molecules to build up larger, more complex ones, such as DNA.
How does this happen?
Wow, you're full of questions today, aren't you?
I'm actually acting so the kids are interested, okay? I already did my MBBS in India.
Okay, back to replication. To answer your question, the enzyme can break down proteins, allowing amino acids to add on to the new molecule.
Okay, so we need to talk about gene expression now.
No, that's the next episode, remember?
Oh wait, so we done?
Yep, bye!
Adios!
Ciao!
Wait, we didn't talk about transcription and everything. Oh, right. That's embarrassing.
So, there is a certain order in which information flows within a biological system. DNA makes RNA, and RNA makes proteins. We talked about how the replication part works, but what exactly are transcription and translation?
Transcription is basically when you take someone's notes and copy them down word for word because you fell asleep during class and no one perfect, right? Because teachers give you so much work that you up all night and you have no sleep. So basically teachers prevent you from doing anything that is life, okay? Like eating, sleeping, pooping, all that kind of stuff, and having a social life, okay?
We touched base on transcription earlier in this episode, but let's take a really detailed look at what happens.
Remember when I said that RNA polymerase binds to the leading strand? Well, RNA polymerase reads the leading strand in order to make mRNA, which is also known as messenger RNA. So your template strand at this point is known as the antisense strand, and the other strand is known as the sense strand.
That makes
Anti-sense, as in, that doesn't make any sense. That was horrible. That was a bad pun. Yeah, I know, I understand this thing, but I just really wanted to make that pun really bad. But, no.
mRNA is formed in the 3 to 5 direction, and before mRNA can go out to the cytoplasm in order to make proteins, it first has to get its introns spliced out. A good way to remember this is that introns are in the way, they need to be taken out. And introns are the non-coding pieces of DNA. Now your exons are the things that remain, and they're regions of RNA that actually code for proteins. Now a poly A tail is added to the 3 end of RNA, and a methylguanine cap is ended to the 5 end. So mRNA then goes to the cytoplasm, and ribosomes and tRNA translate the mRNA into proteins.
So it's like us making a YouTube video. We basically film it out, which is like you composing or RNA polymerase composing the mRNA strand, and then we take out the parts that make no sense and are bloopers and everything. You don't even know about that one. And that are basically introns, okay? Exons are the things that are actually relevant, like the information we give, so they are kept into the thing. The poly A tail, the methyl cap, these are the annotation / description / editing fancy code word.
Wow, that's actually a good analogy.
Oh, really? Thanks, buddy.
Yeah. So the complementary nucleotides to those found on the mRNA strand are brought over by tRNA in order to form amino acids. They carry a special sequence called an anticodon. These amino acids link together and fold with their various polar and nonpolar properties in order to form proteins.
Translation is basically like a school bus, okay? So the driver basically picks out kids from each neighborhood, and each kid can be classified as an amino acid. So it just becomes bigger and bigger and bigger until it becomes full and it's a mess and kids are throwing lunch at each other. But yeah, then you have protein or fully made bus. Yes.
Okay, I think we got everything this time.
Really? Then come here. Thanks for watching Binalogy.
Q&A!